Hi, everyone. Thanks for your patience today. Clearly, we're global health folks and we're not technical folks, so do appreciate your time. I'm Chan Harjavan. I'm honored to host and facilitate this event with Clive and Bruce. While it's always a good time to discuss global health, this discussion is more critical than ever. My background is in global health. Did my master's in public health at Johns Hopkins, and I led the global public health practices at Boston Consulting Group, and then at PricewaterhouseCoopers. I've worked with most global health organizations in the world, whether it be WHO, Gates Foundation, World Bank, UNICEF. I've worked with most of the US government agencies focused on health, and I've worked with most of the pharma companies focused on global health as well. So come with a pretty broad background through my consulting work. Look, through global health, it's facing a lot of challenges today. We saw the devastating effects of COVID. It highlighted the economic impact of COVID, of infectious disease, the socioeconomic disparities. We saw the chilling obfuscation of the Chinese government in withholding health data. We saw some missteps by the WHO, and we saw the perplexing mistrust of science in the US. The ultimate result of this is that we've got a reduction in life expectancy. We need to acknowledge these challenges and some of these failures within the global health community. These challenges being said, our focus study is not to focus on these failures, but to focus on really learning from these two legends we have in global health. In the past few decades, lifespan is essentially doubled, mainly due to vaccines and other global health initiatives. It was 32 years old at the beginning of the last century, and now it's over 72 years. 20 million lives have been saved just with the measles vaccine since 2000, in the last 23 years. It's estimated that 3 million lives were saved just in the US alone with the COVID vaccine developed in a record time in just 6 months. And of course, we're on the verge of eradication of a number of diseases, meningitis A, polio, guinea worm. I think there's 11 cases left of guinea worm. And, with Bruce and Clive, we'll learn from their experiences in managing two tropical diseases, malaria and riverblindness. Malaria continues to affect Americans with almost 1,400 cases annually. That's just in the US, and kills hundreds and thousands in Africa, mainly children. How and why we would be looking at ways to reduce the spread of this disease, both here and abroad. We're going to examine that. We're going to look at onco first, caused by a parasitic worm that threatens 150 million people still in sub-Saharan Africa, where the disease is concentrated. So let me start with Bruce Benton. I'm going to do an introduction of him. He was the World Bank manager of the Riverblindness Control Program from 1985 to 2005, a long tenure. During those years, he led a doubling of the size of the West African control effort and brought that program to a successful conclusion in 2002, having achieved the elimination of the disease throughout a 10-country area and protecting 40 million at-risk Africans from its devastation. In the early 1990s, Bruce organized the launch of the African program for onco control, covering all of the 20 remaining endemic countries on the continent of Africa, and protecting an additional 100 million people from severe disability and blindness. In 2000, the president of the World Bank recognized Bruce's great work with the first ever special presidential award to an individual for leading the riverblindness control effort. After retiring from the bank, he set aside 6 years to travel, interview, and write the book we're going to go through today, Riverblindness in Africa, Taming the Lion's Share, now viewed as being the authoritative account of defeating disease throughout Africa. So with that, let me hand it over to Bruce. He's going to have some slides for us. Then I'm going to introduce Clive, who's going to talk about malaria. And then we're going to open up to question and answer. We'll have quite a bit of time for that. And we're going to go till 4:30 because we started late. So with that, let me hand it over to Bruce. Please take it away.
Thank you, Chan. Good afternoon. Yesterday was the 4th annual World Neglected Tropical Disease Day, NTD Day, a day to draw attention to the importance of addressing these neglected diseases of poverty. Riverblindness was the first of the major NTDs to be tackled 50 years ago. So it's appropriate to review this control effort and how it proved successful to draw lessons that might benefit other NTD control programs. Culling lessons from the riverblindness experience was a major objective of mine in writing the book, Riverblindness in Africa, 2 years ago. I'll touch upon some of these key lessons toward the end of my remarks. Next slide. Here's the agenda that I'll follow. The technical word for the disease is onchocerciasis, or onco, as it's known throughout much of Africa, in part because onchocerciasis is so difficult to spell and pronounce. I'll use onco frequently in my remarks. The layman's term river blindness is a bit misleading because it implies that rivers cause the blindness rather than a parasite. The first program was the West African Onchocerciasis Control Program, or OCP, launched in 1974 to control the fly transmitting the disease. We had no safe, effective drug at that time. Subsequently, the pharmaceutical company Merck discovered ivermectin and took the unprecedented decision to provide it free for as long as needed to eliminate the disease. The availability of free ivermectin enabled us to launch a second program for the rest of Africa, the African Program for Onchocerciasis Control, or APOC, in 1995. I'll cover the vast partnership that was instrumental in the onco success, and finally I'll summarize what I see as the major factors in that success. Next slide. When the first program began, approximately 150 million people lived in at-risk areas in 31 African countries. 99.6% of the world's cases were in Africa. The disease also existed in the Americas, brought over from Africa by the slave trade, and in Yemen. But the disease in these areas was only a drop in the bucket compared to Africa. The red and black areas on this map show where the disease had reached crisis levels as a public health problem. Next. The two major manifestations of onco are blindness and skin disease. This photo shows an eye blinded by microscopic worms that have entered and died off in the eye. The average age of the onset of oncoblindness is 30 when victims are in the prime of life. Next. The word for onco in Bambara, the largest ethnic language in Mali, is Mara, which also means lion stare. Those blinded by the disease have a fixed, opaque stare resembling a lion's gaze. This slide shows, back please. A common scene throughout sub-Saharan Africa has been young children leading onco-blinded adults around by sticks. Hence, the disease has also prevented young children from attending school in order to take care of their elders. The photo in the upper right from South Sudan was chosen by Johns Hopkins University Press as a cover for my book. Next. Children also wind up leading long lines of onco-blinded adults. Two classic photos here, one taken in Chad, a decade before the control effort began. Next. Unsightly skin lesions and unbearable itching are caused by microscopic worms continuously migrating beneath the surface of the skin. The lesions sometimes result in victims being ostracized, and the incessant unbearable itching can lead to suicide. In terms of disability-adjusted life year, or DALY calculations, oncoskin disease, notably the itching, is a greater health burden than oncoblindness. Next. Another skin manifestation is oncodermatitis, known as leopard skin, that causes depigmentation and uncontrollable itching, usually around the lower legs. Next. This photo shows the adult female worm, measuring 2.5 feet long. It is usually found curled up with a shorter adult male worm in stationary nodules around the waist area. The female worm has a reproductive lifespan of about 12 years and continually produces millions of microscopic offspring, known as microfilariae, which fill up the body and do all the damage. Next. The man on the left has 4 pairs of adult worms in nodules. The young boy on the right, about 8 years old, already has an adult worm nodule. He is likely experiencing itching, and without treatment, he'll have lesions covering much of his body in 10 years and diminished sight approaching permanent blindness in 20 years. Next. On the right is the black fly, Simulium damnosum, that transmits the parasite from person to person. This fly can travel up to 300 miles with the wind currents. On the left is the pupa of the fly that attaches to sticks, stones, and leaves in fast-flowing rivers. Vector control involves killing the black fly pupa with insecticides, screened and cleared by an independent ecological group as environmentally safe, to prevent the fly from propagating, biting, and transmitting the disease. Next. Under the West African OCP, we employed a fleet of 12 helicopters and 1 fixed-wing aircraft to spray up to 30,000 miles, of rivers a week throughout the West African 11-country area, 3 times the lengths of the Mississippi, Missouri, Rio Grande, Colorado, Columbia, and Ohio rivers combined. It was a huge operation and became the largest inter-country health program in the world by the late 1980s. Next. The program employed several hundred flycatchers around the 11-country area to catch and dissect flies to determine if they carried the parasite. If so, data would be relayed via radio to program headquarters in Ouagadougou, Burkina Faso, and then on to the helicopter pilots to guide them on where to concentrate their spraying. Next. This shows prevalence throughout the West African 11-country area when OCP began. Most countries had prevalence rates of 60% or greater, the most heavily infected area known anywhere in the world at the time. Robert McNamara, president of the World Bank, launched OCP in 1974. He visited Upper Volta, now Burkina Faso, in 1972, and saw the disease firsthand. He became convinced that the World Bank had to lead a control effort. Otherwise, there was little prospect for development in that whole subregion of West Africa. Launching OCP was bold and risky. The World Bank had never supported a health initiative up to then and had no staff with expertise in health. Furthermore, the control effort had to be regional rather than country by country due to the long flight range of the black fly, thus requiring grant financing rather than bank loans or credits. Under World Bank procedures, a loan or credit had to be guaranteed by a borrowing country. In this case, there was no individual country to borrow and provide the guarantee. So grant financing had to be mobilized by the World Bank from a wide range of bilateral donors to fund the control effort. Also, the old guard in the World Bank opposed McNamara's initiative. In their view, health interventions would not promote development as effectively as projects in the so-called productive sectors, like transportation, industry, and agriculture. In the end, the old guard would be proven wrong about this. When I was recruited to manage the effort for the World Bank in early 1985, the program covered a 7-country area shown by the solid black line in the right half of the map. My principal tasks were to chair the Committee of Sponsoring Agencies, a steering committee that oversaw program operations consisting of the World Bank, WHO, UNDP, and FAO, and to mobilize all donor funding. At the time, we faced a serious crisis known as re-invasion. Flies were becoming infective to the west and the southeast and invading that original 7-country area. Hence, it was impossible to bring the program to a successful conclusion unless we could extend vector control operations to the west and southeast to cover the limits of the breeding sites of the black fly throughout the subregion. The expansion entailed increasing coverage from 7 to 11 countries, from 25 million to 40 million people, and doubling the control area to 3 times the size of France. The expansion wound up costing an additional $200 million and took a decade to complete. Next. At the completion of OCP in 2002, the disease had been largely eliminated throughout the subregion, with the exception of Sierra Leone, which experienced civil war throughout the 1990s. That war prevented control in Sierra Leone for a decade. Fortunately, the disease was later eliminated there through ivermectin treatment, supported by the follow-on program, the African Program for Onchocerciasis Control, or APOC. Next. Successful control in West Africa opened up 250,000 square kilometers of arable land that could not be inhabited or cultivated due to the severity of the disease. That swath of land, which was larger than Great Britain and covered parts of 10 countries, was spontaneously resettled and cultivated following control, enabling those countries to transition from importers to exporters of agricultural products. 2 countries, Burkina Faso and Mali, became Africa's largest cotton producers and experienced GNP growth rates surpassing 10% throughout the 1990s. We carried out cost-benefit analyses in the 1990s, which concluded that the West African program had an economic rate of return of 20%, surpassing many of the World Bank's most successful projects, regardless of sector, and demonstrating that health projects can be powerful in catalyzing development. Next, a highly effective drug, ivermectin, was discovered and became available in the late 1980s. Dr. Satoshi Omura, senior scientist at the Japanese Kitasato Institute, shown here, found an organism appearing to have promising bioactivity in a soil sample on a golf course in Ito, Japan. Based on an agreement between Kitasato and Merck, Omura sent the culture to Merck to be screened for antiparasitic activity. This photo is a reenactment of Omura finding the culture in a soil sample on that golf course. Next. Subsequently, a senior Merck scientist, Bill Campbell, shown on the left, screened the culture and discovered the ivermectin molecule and later its effectiveness against onchocerciasis. I was fortunate to spend hours interviewing Bill for the book and to learn the fascinating story of how that molecule was discovered. It became perilously close to being missed entirely because the only mouse used to screen that culture nearly died prematurely because of a toxic impurity in that particular screening. If it had been missed, it's quite possible that the miracle molecule, as it has later become known, might never have been discovered. The exact culture in the soil sample from the Japanese golf course has never been found anywhere in the world since. Merck's subsequent clinical trials showed that 1 dose of ivermectin killed off 95% of the microscopic worms in the body with virtually no side effects, although it did not kill the adult worm. So ivermectin would have to be given at least annually for the roughly 12-year reproductive lifespan of the adult worm. This photo was taken at the 2015 Nobel Prize in Medicine ceremony in Stockholm, where Bill Campbell and Satoshi Omura shared the Nobel Prize for the discovery of ivermectin. Next to Campbell is Merck CEO Roy Vagelos, who took the unprecedented decision in 1987 to donate ivermectin, or Mectizan, the name of the human formulation of ivermectin, for as long as needed to as many who need it to eliminate onco. Vagelos had concluded that Merck would be unable to sell the drug due to the poverty of the endemic countries and the affected populations, and that it would be unethical to withhold it for such a devastating widespread disease. Vagelos's unprecedented decision stunned the pharmaceutical world at the time. There was concern that donating a drug for a widespread disease would undermine research and development for new drugs for other diseases, notably the neglected tropical diseases. However, the opposite happened. Other pharmaceutical companies proceeded to develop drugs to treat the other major NTDs and to donate them, in part to achieve the highly positive public relations that Merck benefited from its ivermectin donation. Next, the question then became how to get ivermectin, or Mectizan, to a high enough proportion of the infected population to halt oncotransmission, which required reaching at least 65% of the infected population. 2 years of operational research determined that a novel delivery method called community-directed treatment could reach 70% and 80, between 70% and 80% of the infected population, well above that 65% threshold. Under this approach, the community took full ownership for treating its members, including devising and implementing the distribution strategy, keeping records, storing the drug, and following the Merck protocol for safe treatment. Many, including Merck, thought relying on the communities wouldn't work because most community distributors were illiterate volunteers. But the approach far exceeded expectations, and eventually, community-directed treatment became the principal method to distribute other NTD drugs in the rural areas. Next, these maps show declining prevalence of onco over time. Next, by 2015, the disease had largely disappeared from West, Southern, and East Africa. It remained concentrated primarily in Central Africa, notably Congo-Kinshasa and South Sudan, where ivermectin treatment was impeded by civil unrest. Nevertheless, ivermectin delivery continued by community-directed treatment with free ivermectin. Prevalence continues to decline. Next, please. And the disease is projected to disappear as a public health problem over the next several years. Next, I work closely with 5 directors of OCP and APOC based at program headquarters in Ouagadougou, who were hired by WHO as the program's executing agency. They were highly effective managers and skilled scientists, medical doctors, ophthalmologists, and entomologists. Each was driven by a strong sense of compassion for the poorest of the poor suffering from the disease. I can't say enough about their hard work, dedication, and determination to wipe out this disease. Their leadership in achieving results on the ground was central to the onco success. Next, a vast global partnership powered the onco programs. 7 partner groups with differing comparative advantages functioned synergistically to achieve and sustain control. First, you had the 4 sponsoring agencies, WHO, World Bank, UNDP, and FAO, making up the steering committee overseeing operations. Second, you had the 31 African governments. The most heavily infected countries were Burkina Faso, Nigeria, and Congo-Kinshasa. Third, 27 donors, countries, international organizations, and foundations around the world. The largest donors were USAID, the World Bank, and the government of the Netherlands. Fourth, 35 NGOs supporting the local communities in achieving comprehensive treatment with ivermectin. The most active NGOs were Sightsavers and the Carter Center. Fifth, Merck, responsible for providing ivermectin to the programs. Sixth, tropical disease research entities, including Johns Hopkins University. Hopkins has been the most active of the research universities in the control effort. A team from Hopkins carried out the critical phase 4 ivermectin trials in Liberia, and a Bloomberg School professor, Gilbert Burnham, has chaired the Mectizan Expert Committee set up by Merck to monitor ivermectin treatment for nearly a decade. And he led the final evaluation of the second onco program, APOC, in 2016. And last but not least, the 150,000 local communities in the endemic countries responsible for community-directed treatment and ensuring comprehensive coverage of the at-risk populations. Keeping this vast coalition together and thriving over decades was instrumental to the onco success. This was achieved in part by ensuring that the program objectives were clear and consistent over time, so that all the partners were on the same page and pulled in the same direction, and by continually consulting each partner on program progress and its specific role in achieving the objectives. The program director and I, for example, visited every donor annually to discuss program challenges and the level of that donor support required to stay on track in achieving elimination. Next, one interesting symbolic device employed to help hold the coalition together was the Riverblindness statue. Alaskan sculptor R.T. Wallen, fondly known as Skip, sculpted the small statue on the left of a young child leading an onco-blind adult in the early 1990s. Based on that statuette, Skip was commissioned to sculpt an 8-foot-high, larger-than-life-size bronze statue shown on the right in Skip's Juneau, Alaska studio. Through that mold, we wound up producing 6 nearly identical statues, which were donated to be prominently placed at the headquarters of major partners in the program: Merck, the World Bank, WHO Geneva, the Carter Center, the government of the Netherlands, and Lions Club International. Next, during an onco meeting in 2001, I took this photo of Robert McNamara standing in front of the Riverblindness statue in the atrium of the World Bank's main building in Washington, D.C. McNamara had retired from the bank 20 years earlier. The onco program was one of his proudest achievements as World Bank president. During his retirement, he invited me to his nearby K Street office from time to time to discuss the program's progress and to reminisce on why he felt launching it was so important. Next, the career high point for me came in 2000 when World Bank President James Wolfenson presented me with a special presidential award for leading the onco elimination effort throughout Africa. Subsequently, Jim wrote the foreword for my book shortly before he sadly passed away in November 2020. Along with McNamara, Jim Wolfenson was the most active and strongest supporter of the onco programs among World Bank presidents. His involvement and outspoken support were critical in holding the coalition together and in encouraging full-fledged donor support for the effort. Next, these are what I regard as major factors for the onco success. First, long-term commitment. The coalition held together for 43 years. The original donors stayed with the program for an unprecedented 35 years. Second, comprehensive approach. No endemic country or at-risk community was left uncovered by control operations. Although civil unrest occasionally inhibited control in some areas. Third, cost effectiveness. The annual cost of program operations, whether vector control or drug treatment, never exceeded $1 per person protected per year. Fourth, substantial economic development and humanitarian benefits. Dramatic increases in agricultural production and in human productivity, coupled with greatly reduced human suffering, were highly valued by the donors, the African governments, and the local communities. These benefits were powerful inducements to continue supporting the programs for more than 4 decades. Fifth, clear, consistent objectives over time were important in keeping all partners aligned throughout the effort. Sixth, treatment buy-in. Vector control and ivermectin treatment were extremely popular with the affected populations, thereby ensuring strong compliance at the country and community levels. Vector control reduced the pain and nuisance of incessant fly biting in many rural areas, and ivermectin alleviated the unbearable itching within 3 days, leading those infected to line up enthusiastically to take the drug. Seventh, ownership by the affected communities. Through community-directed treatment, the communities actively assumed responsibility for treating their members with ivermectin, thereby achieving high enough treatment levels to block oncotransmission. And finally, 8, the regional multi-country approach. Going regional was necessary due to the long flight range of the black fly, but it had considerable advantages in ensuring comprehensive control and in promoting cross-country collaboration in addressing the disease. Thank you, Chan, for your attention to this, and I look forward to the discussion period and answering any questions that the audience may have. Thank you.
Yeah. Yes. No, thanks, Bruce. And yes, please post any questions that you may have on Bruce's talk or any other public health topics in the chat room. We'll aggregate them, and after Clive speaks, we'll have a Q&A on that as well. So we'll be able to see your questions, and we'll be able to ask both of them questions and have a bit of a dialogue here as well. So thank you, Bruce, for that wonderful talk. It's just amazing what you've done, and it's nice to connect that with the Hopkins community as well. Next, we have Dr. Clive Schiff, a fellow Southern African. I was born in South Africa. Clive was born in Rhodesia, but then studied in South Africa. He completed his master's in science and zoology and entomology in 1956 and did his PhD at Rhodes in 1964. He now serves as a professor at Hopkins within the molecular microbiology and immunology department. He's been a key member of the teams that have crushed the tsetse fly as a public health threat in South Africa. When he was young, as a member of the, as a field officer and young researcher, he helped develop several life-saving innovations, including pit latrines and environmental interventions for village water supplies. I think that's really important in understanding the environment and other health interventions in saving lives. In his current role at Johns Hopkins, he's led key research into the relationship between schisto and bladder cancer, and remains an investigator at the Malaria Institute in Zambia, in Maka. And he functions as a core faculty for the research community at Johns Hopkins with a focus on field research. Again, I think his local viewpoint, I think, is really, really important. It's very Africa-centric and looking at parasitic diseases in Africa. He also contributed to a number of research findings, including the diagnosis of both the African species of schisto by detection of the specific DNA fragments from urine samples in rural health centers, communities, and malaria case detection in Zambia using mobile telephones as a means to detect potential reservoirs of infection in unstable transmission conditions. Sensitivities and specifications of diagnostic tests and infection prevention of schisto estimated from data on adults in villages northwest of Accra, Ghana, and PCR detection of the Plasmodium falciparum parasite in human urine and saliva samples. So that was a lot, But let me hand this over to Dr. Schiff to talk more about his work.
Thank you. Thank you very much indeed, Chan. I didn't realize that we're from the same country. So I'm going to talk about malaria, and I suggested that we use neglected tropical diseases as let me just get rid of these pictures. Neglected tropical diseases, because although malaria is not is not proposed as a neglected tropical disease by WHO and other organizations, it actually is neglected because the funds that go to studying malaria really don't go to the tropics. They don't go as to the endemic countries in sufficient quantities to reach the same kind of success that we've seen just recently now with ivermectin and the Onchocerciasis control program. That was highly specific and focused. Malaria, in fact, is very, very widespread, and I will get onto that just now and show you how global it has been. And it is, and that is one of the reasons why there's so much interest in the northern and western countries. But in modern times, in the last 40 or 50 years, it has been concentrated mainly in Africa and Southeast Asia and parts of India. And we will get onto that as I go through the story. But in fact, I'm being at Hopkins and seeing malaria and visiting the American Society of Tropical Medicine and Hygiene meetings on a regular basis. I've come across and know most of the groups working on malaria in Africa and elsewhere. So I've seen what goes on. I myself started working, as Chan said earlier on, in what was Rhodesia in those days, but now Zimbabwe, with the Ministry of Health, which had an interest in controlling malaria and other tropical diseases, particularly bilharzia or schistosomiasis, which is still highly prevalent in Africa. So the point I want to make here is that the research being done in the developed countries, in my own department at Hopkins, is very much focused on the parasite itself, but not so much on the people who suffer, on the disease. And what we have to do really is to make sure that adequate training of scientists, local scientists, African scientists, are funded so that they can work on the issues, the malaria issues in their home countries, instead of migrating to Europe and the United States and taking up positions in universities away from their homeland. Next slide. I'm going to talk to you now about the life cycle. So could you click it again? The life cycle of this parasite is very complicated. The next slide will show you even more accurately the processes of this parasite. This is a horrific slide, and I apologize, but I will point to several important aspects of the parasite. The first one is it's transmitted to humans by a mosquito, not any mosquito, but very mosquitoes belonging to the genus Anopheles, and there are about 400 members of the genus Anopheles, but only a small proportion of that, probably about 10 or 12 species, will actually transmit malaria. So they're very specific. And we'll talk about that in due course. So the malaria parasite is injected into the human being when an infected mosquito bites. And if you look on the top right-hand side, it says liver stage, liver cell, and liver stages. The parasite migrates into a liver cell and replicates prolifically in the liver cell. The malaria parasite, one parasite will produce several thousand what we call merozoites, which are zoites from the malaria parasite. Each one is an individual parasite, and when the cell bursts after about a week, these penetrate into the red blood cell. And that is the cycle on the bottom right. It says erythrocytic cycle. You don't have to worry too much about the different colors there, but the cycle for the falciparum, the main parasite in Africa and most of the world, as a matter of fact, produces about 20 to 24 merozoites after a 48-hour cycle. So every 48 hours, the red blood cell disintegrates and the merozoite emerges. These merozoites cluster in red blood cells very rapidly. Colleagues of mine working in my department have shown that actually the merozoite emerges from a parasite, from a red blood cell, and invades a new red blood cell in a few seconds, something like 12 to 15 seconds. So there isn't much time for the immune system of the host to attack the parasite, because inside the red blood cell, the immune system is not active, it can't penetrate into the cell, there's no nucleus in the cell, and the parasites will replicate again. So the problem goes round and round. At some stage, sexual stages of the parasite occur, and these remain in red blood cells. And when a mosquito bites the infected person, they ingest a male and a female form of the parasite, which then replicate inside the mosquito. So actually, the mosquito is the driving force of malaria in a population. Next slide, please. this slide gives you an idea of the extension of malaria in early days when humans didn't have the appropriate drugs to treat it, or didn't even know what was going on. But if you look at this map of the world, you'll see areas that are gray and areas that are pale, light green, dark green, and very dark green. And the very dark greens are what we call holoendemic, where annual transmission of malaria is still going on continuously, so that you can get malaria at any time of the year in the areas of the holoendemicity. You will see they're what are there are primarily in Central Africa these days. If we look at the top of the map and see that light-colored area, which is in Europe, Asia, Europe, and spreads across to North America, that is what they call epidemic malaria. And in the 19th century, there were huge outbreaks in different parts of the world in that epidemic area. In North America, and certainly in North America and South America, malaria really didn't exist in the Western Hemisphere until migrants from Europe settled in North America and South America, and then when people were brought from Africa, and they also brought more parasites, so the malaria was introduced to the Americas much more recently than it had been in the main, in the rest of the world. So you can see now that there are still large areas of territory within the black margins that are on this map where malaria transmission is still endemic, and where people die in large numbers. And it's not only the fact that malaria kills children, it also kills adults, but not to the same extent. And we do have drugs now to save lives. So really and truly, malaria shouldn't kill anybody if the proper drugs and the proper organization is in place in the endemic areas. Anyway, this is just a bird's eye view for you to look at, to get a perspective of what it was and why it's of high interest globally. Next slide, please. So I wanted to I wanted to focus more a little bit on the mosquito itself, because mosquitoes transmit malaria. This is the driving force of malaria. The mosquito, in the sexual transmission, the sexual stage of the parasite occurs in the mosquito. The male and female gametes form there and proliferate and produce large quantities of infective forms of the parasite, which migrate into the salivary glands of the mosquito. That process takes about a week in the mosquito. And then when that mosquito feeds, when it salivates, as it probes the skin, the infective forms of the parasite get into the host, into the human, and start to proliferate in the way we've just seen before. Next slide, please. In my work, I grew up in Zimbabwe and worked for the Ministry of Health, and we had a malaria control program which started very soon after the Second World War, when the insecticide DDT became available. It was a tool that was found effective by spraying in the houses where people slept at night, spraying the bedrooms, the walls, because the mosquitoes, when they feed on you, they are nocturnal. They feed at night. They don't feed during the day. And when they ingest blood, their body weight increases considerably. Can we go back to the mosquito? I don't know if we can go back. Okay, well, no, back, not forward. Yes. I want you to look at this. You see, this mosquito is feeding, but it's also excreting water, fluid from the blood meal. These mosquitoes, when they fill with blood, become the mass of the mosquito probably triples its weight. So it can't fly far. So having fed, they will fly onto the wall of the house. And if there's insecticide on the wall of the house, the mosquito picks up the insecticide and dies. That was highly successful, and I will talk about that a little later on, but I just wanted you to know that this is the target of very much if the way to control malaria is to control the parasite, because there are major issues about trying to remove parasites from human beings. Okay, next one. And so, no, let me just go back, because I spent 5 years working in Tanzania, and this is in a city north of Dar es Salaam called Bagamoyo, and there are a series of boarding schools there, and we collaborated with the Ministry of Health and did surveys in the school children. And these children came out of the classroom, but we found that 70% of those were parasitemic. They actually had malaria. So can I go to the next slide? You'll see the proportion of people, of children at different ages from that community who were carrying malaria parasites in what we put down as asymptomatic, because the children were at school, they weren't suffering, but they were all infected, and and affected by the disease. But measuring the effect of malaria on that population has really never been done. It's easy to treat. It's simple. There's several drugs. Chloroquine was the good one in Africa at this particular time. But this is an example of the extent of the infection in people who live in the field. I mean, they get exposed to mosquitoes. So something really had to be done about it. And that was the next stage, or what we had actually implemented in Zimbabwe when I was working there. Can you go on? Next slide. So what I would like to do now is to move to a situation similar to what we found in Tanzania, but this is in Zambia, where where I've been working for some 25 years now, because the Bloomberg Philanthropies decided to support malaria work at Johns Hopkins School of Public Health, and we built a facility in rural Zambia in the southern province, where there's an H that is a hospital, but it's also a research station, which is highly, it's a large research institute now. It has a staff of several medical doctors and a PhD, all African trained, and doing research, which I'll talk to you a little bit about as the subject goes on. What one has to focus on in the rural situation in Zambia, is typical of many parts of Africa, is people live on farms, distributed all over the country. They're rural, rural conditions. They live on farms in small compounds, housing compounds on the farm where the laborers and the owner lives. And so there are something, the area in this map is about 5,000 square kilometers, and each of those crosses with the names attached are clinics that are run by the Ministry of Health. What's important in Zambia is that they have a very strong Ministry of Health, which is dedicated towards controlling malaria. So these clinics are well supported. The staff are paid properly, which wasn't always the case in Tanzania. And this whole area is agriculturally developed. The colors here are really from altitude. So the green areas are near a major river system that runs just at the north of this. And you can see the blue lines and the white lines are river drainage systems. But the population here is generally agriculture. There aren't any big cities, towns, or even big villages. There are, but these clinics are maintained by the Ministry of Health. And one of the studies that we did over a period of about 5 or 6 years was to study the prevalence of malaria in these different areas. So could I have the next slide, please? So we divided that up into the floodplain, which was the green part, a transitional zone, which was a a little higher, the floodplain about 900 meters above sea level. And then the brown area was about 1,100 meters above sea level. And what we put here as heartland was about 1,200 meters above sea level. So it's a fairly, it's not hilly countryside, but there are different heights where people have put their farms. And what we did was we collected from all of the all of the clinics every week the number of malaria positive diagnoses that were carried out the week before. So we plotted that information on this map, on this graph here. And what you can see on the left, it starts off, the one, of course, is the first week of the year, and that's in the middle of summer. The rainy season in Zambia starts around about week 42, 43. It's in November, and then it goes on until about week 20, 19 or 20, when the rains stop. And if you look at the graph, you can see that in the floodplain there was transmission in transmission of malaria in the population living in the floodplain. Was these were people who came to the clinic sick and were diagnosed by the staff before treatment. But you can see that in the transition zone and really in the heartland, there was very little transmission from week 27 till week 47. So for that period of time, it's cold. It's cold and dry, or cool and dry. So there are definite patterns and different areas where there's more transmission, and you need to know this when you just lay out a program to control malaria extensively. So in the next slide, please. So what I want to show you here is what we do under those circumstances to try and prevent transmission, to prevent people getting infected. So this is how you spray with a residual insecticide. This is not DDT. It's in a number. There are standard insecticides that are used for spraying on the walls, but there's a lot of equipment that's required. First of all, the insecticide has to be weighed out in bags. So there's 100-gram bags, and each bag is sufficient to put into one of the spray cans and fill up the spray can, and then you shake it up, and then you spray that on the wall of the house. Next slide. People have to be shown how to do the spraying. It's not something that you could just pick up and do it. The spray has to be done so that you wet the wall, but the wall doesn't run, because you don't want all the insecticide to run down to the ground, because when you spray somebody's house, it's got to stay on the wall, because that's where the mosquitoes are likely to sit after they've had a blood meal. So there's a lot of training involved, and there's also a lot of transport expenses, because all of these people have got to be carried from one house to another with all the equipment that they use when they do indoor residual spraying. Okay, next slide. However, And this was one of the things that we were examining when we worked in Tanzania. There was a group in the UK that suggested that if one could treat mosquito nets with an insecticide, then the mosquito would be exposed to insecticide when it's probing for a host, but and if it gets through and bites the host, it will pick up insecticides, and if it gets infected, it probably would not survive to be able to transmit the infection, because the mosquitoes have to feed every 2 or 3 days, and if they come back to a treated bed net after 2 or 3 days, they will pick up more insecticides, and it should kill them off. And it has been extremely effective. So the next slide. So WHO produced a manual about insecticides, and this was an illustration that I copied from it, which gives you an example of how these insecticide-treated nets can protect not only people sleeping under the net, but people sleeping outside the net, because the mosquitoes are not aware of the insecticide on the net. Even if there are holes in the net, they have to find these holes, and so they probe around until they get it through a hole and then can feed on somebody. But also they're picking up insecticide, and treating the mosquito net with an insecticide was really a major change from indoor residual spraying, because these nets were dipped in insecticide, or more recently, the actual mosquito net fibers were impregnated while the fiber was being manufactured, impregnated with insecticide, so that the mosquitoes, so that when you've got a net made up, the insecticide permeates out onto the outside of the fiber, and when a mosquito probes, it picks up insecticides and dies. And those insecticides are actually derived from a plant called pyrethrum, and so these insecticides are called pyrethroids. They are stable forms of pyrethrum. So there are 2 different ways to control malaria, which have been done, and are still the only way we can reduce transmission of malaria in the endemic areas. So there's a lot of work that has to be done, and it requires a well-established Ministry of Health with scientists to control the use of insecticides, to measure, to make sure that spraying is done properly, to make sure that the nets are still active. So there's a tremendous amount of surveillance necessary. Can I have the next slide, please? Recently, I thought I would just give you a little bit of information about a recent experiment that we did in Zambia. We worried about the hot, dry season and how do mosquitoes survive, because there is transmission in these houses in the hot, dry season. not much, but there is. And we measured humidity in the trees, right? That are, most people in Zambia, when they build a house, they build it near a tree because the tree gives them shade and keeps the place cool during summer. So the device that's in this pot at the top, the gentleman has just put in place, is a thing called a logger, and it measures temperature and relative humidity every hour. And so you get information about this. And I'll show you towards the end some data that shows how significant the moisture in the trees is for transmission that occurs in the hot, dry season when there's no rain, but from moisture that actually comes from the trees. One must remember that in the tropical areas, trees do not shed their leaves completely like they do in the colder countries. So in the tropics, even if the ground is dry, there's not much water in the rivers, there's sufficient moisture in the trees. And when the air is stable, there's sufficient moisture to allow the mosquitoes to feed. Next slide, please. Here's some data from that paper showing the red bars are the incidence of malaria in the clinics near each tree that we talked about. The blue lines represent rainfall, and the green line at the bottom represents the temperature, and the pale blue, which is in the middle, is relative humidity. So there's very high relative humidity. This is over 3 different years. So the pattern is the same year after year after year. Because the beginning is 2017, the middle is 2018, and the decline on the right-hand side is 2019. So these data were quite useful and show that mosquitoes are vulnerable, are able to increase the case incidence rate in the clinics when there's periods of time where there's this moisture and sufficient moisture for a period of time, like 3 hours. Next slide, please. So this is these data have just recently been published, and you can see as you move from the left to the right, these dots, which are the weekly incidence of malaria cases in the clinics, and not only, and the dot is the nighttime count of a measurement that lasted for 3 hours with a 50% relative humidity. So if the relative humidity was 50%, for 3 hours, then there's more chance of malaria being transmitted over those 3 years. So it's interesting, and it gives you an idea how transmission can occur even in the hot, dry season. Next one. So I want to sort of end up here talking about vaccines, because there's a lot of effort being made. In fact, really huge amounts of money are being spent on a vaccine. Either the PSF 5066 was one of the first, but it was a failure. And then there's a group in DC working on developing, expanding live sporozoites. These are from infected mosquitoes, which you can inject into. You can you can't freeze them, but you can. If you inject those into a person and then expose the person to malaria, you get protection. This was first done in about 1950 by a friend of mine when he infected himself, and first of all with liver sporozoites, which had been irradiated with X-rays and things, and so they were not viable, but they were still active. And but it's still very much in the air. The RTSS at the bottom is a vaccine that has been approved by WHO, but the vaccine requires 4 different doses at 1 month apart. 1 month apart. So you give an initial inoculation to a person, and then a month later a second, month later a third, month later a fourth, and that gives something like 30 to 35% protection for 6 months. So it's not really a very effective vaccine, but it is the only one that is available. And in testing this vaccine in different parts of Africa, they spent over $500,000 just doing the tests. It's very expensive to work on vaccines in malaria because, as I showed earlier on, the parasite is not very vulnerable to a vaccine because it's killed off very quickly. The parasite goes from one red blood cell to another very quickly, and so it's not really exposed to the effect of the vaccine effectively. So we're really suffering from that. So can I have the last slide? So I just wanted to put a statement in which I met this gentleman, Socrates Litzios, who worked for WHO for about 20 years, and who wrote this in 2001 about malaria control. And really, he says malaria control depends on the presence of essential public health functions. And health services in tropical countries are not adequately developed as a public health capacity adequate to control malaria. The problems are sustainability, maintaining personnel, and focused on malaria control. The work has to be done in the field, and it has to be done by local scientists, not expatriates who go to the field and collect specimens and then take them home and work on them. So this is really I suppose the take-home message for what we've shown over the years. You have to have a good health system in place, and this health system has to be supported by locally trained scientists who understand the parasite and its epidemiology. So really, that's what I wanted to say, and I think from what I've been talking about is you can see that it's still a neglected tropical disease, because in most of the tropical countries, the conditions are not available to meet this quality of need. Thank you.
Yeah, thanks, Clive. That was wonderful. So we do have a few questions, and I said earlier we can go on longer as well. We can go right up till 4:30 if questions keep flowing in. The first question, and I think it's to Bruce, but I think Clive, I think you may have an opinion on this as well. But let me read it off. It's from John Gibb. It says, APOC ended in 2016. How has progress been since then? And what are the key challenges to control and elimination? And how do the endemic countries contribute and in what ways? So this really is a direct follow-on from what Clive said was, the local health system infrastructure, the role that plays, the role that these countries themselves play, the governance, et cetera. And Bruce wanted, to see if you had a thought on that. And a side question for you, Bruce, is why did the World Bank take the lead with you on riverblindness versus WHO or even the countries themselves? So let me have Bruce go first, and then we'll have Clive jump in as well. So.
Yeah. Okay. Thanks, Chen. So the intention had been to actually extend APOC. It closed at the end of 2015. The intention had been to extend it for another 10 years. I think John Gibb knows the story, and for a variety of reasons that never happened. Fortunately, the infrastructure put in place by APOC in terms of the community-directed treatment set up with support from NGOs enabled the countries, enabled distribution of ivermectin to continue. There was some concern that COVID would stop that. That didn't happen, although it did interrupt it to some extent. And what happened was that as a result of new drugs becoming available from pharmaceutical companies for the neglected tropical diseases, one program was established in WHO, the Regional Office for Africa, covering all of the 5 major neglected tropical diseases, and to get those drugs out to eliminate them. That's then the approach being pursued now. And you have the 5 are onchocerciasis, lymphatic filariasis or elephantiasis, schistosomiasis, trachoma, and soil-transmitted helminths. Now, drugs now do exist, in part because Merck came up with a free drug for onco, and then subsequently these other pharmaceutical companies came up with drugs and donated them. They're now being used to try to eliminate these diseases through this program, this new program at WHO in the Regional Office for Africa called ESPN, or Expanded Special Project for the Elimination of Neglected Tropical Diseases. I would expect that onco is going to be totally eliminated over time. We know with ivermectin we can stop transmission. It's a matter of getting it out to the infected populations, and there are other diseases that are coming along similarly, especially for lymphatic filariasis and sarcoma. And so your question, Chen, was about why did the World Bank take the lead on this, on onchocerciasis back in 1974? And first of all, Robert McNamara was pretty determined. He jumped right in there. He felt this had to be done. Otherwise, you couldn't. I mean, the World Bank's mandate was development, and he couldn't pursue development in that in a large part of Africa unless you could somehow find a way to bring this disease under control. The other thing was that at WHO at the time, you had a new Director General, Wolfgang Mahler, who came in 1973 before OCP was started, and Mahler was pushing primary health care, horizontal programs, not vertical programs. He did not like the single disease programs, and he wasn't all that interested in pursuing this. So the World Bank sort of stepped in to fill the void. And the other thing is we didn't have a drug at the time. And I think WHO felt less comfortable pursuing a vector control strategy than it would have been had there been a drug, but there was no drug at the time. So the World Bank stepped in, filled the void. However, it was very much of a collaborative program between the World Bank and WHO.
I think part of it, and I'll transition to Clive here to maybe, Bruce, you have more comments, but then to Clive to answer the question as well, but I think part of the question was around country ownership, especially for endemic countries. How are they prioritizing this? How are they, where are the funds coming from, and are they contributing funding as well? This would be both for riverblindness and then Clive, for you, same question for malaria.
Well, so the ESPN program, which has responsibility for these 5 major vector tropical diseases, is mobilizing funding to support this, and the in-country infrastructure that I know the best is the community-directed treatment approach through the communities, and they're interested and active, and they are also taking responsibility for some of these other diseases as well. I think what we lost when APOC came to an end and ESPN started up, we sort of lost that regional focus. And as a result, you have these cross-border areas which are particular weaknesses with the ongoing program effort. And it's not so easy to get countries to collaborate in those cross-border areas to reduce transmission of a disease. And it's particularly important for onchocerciasis because many of the rivers form the borders of these countries. And that's where the flies are breeding.
Oh, that's interesting.
Then picking up and getting infected and transmitting the disease. And you really need to focus on those cross-border areas. And I think that's a weakness right now with the special program. With the
Clive, yep, same question for you, but for malaria, country ownership, funding, et cetera.
Well, that is precisely the problem. I worked in Zambia. They have a very good health infrastructure. The Ministry of Health is very orientated towards malaria control. Yet in the northeast of Zambia, in an area called Nchilengi, which is very swampy, the malaria programs have continued with all the insecticide treatment that has been. Something's wrong with my screen here. Sorry, let me move. Yeah, I'm out of the way.
I think you're fine. I see it too. You're fine.
It seems to be That's not me talking. It's just
No, I would just keep going, Clive. I think we can see you fine.
Okay.
Yeah.
So the issue in Zambia is that the funds come from the president's Malaria Initiative, which is USAID. The USAID programs are passed to companies that put together a team that will be in the field and in the endemic country, and then the team in the field, which has got, say, one or two American scientists supervising it, will subcontract to a local company that does the spraying. And you can't be sure that the actual spraying is done appropriately. And one has to realize these insecticides have commercial value, and the spraymen get paid a very small amount of money to do the work. And so the local farmers would pay them to give us some of the insecticide, and then you don't have so much to spray on the walls. I have seen that happen. I've seen that happen in Zimbabwe. Well, certainly we found it in Rhodesia, and we had to supervise. Even the spraymen had to be re-supervised by scientific people who, people with a scientific background, so that they could make sure that the spraying actually had the insecticide in it. We could test for that. So there's a whole infrastructure in place that you have to have functioning all the way down from the top to the bottom. And it's very difficult to sustain that sort of thing in most African countries, because the governments, the government personnel like to keep the money for themselves.
I grew up in Durban, and malaria was essentially eliminated in Durban, as you know,
yes,
because it was a stable, and it is still for now at least a stable government, so they can control it.
Oh yeah,
it's just quite endemic.
Yeah, and they've got plenty of scientists there. They've got universities there that go and do the work and can supervise, can check. But if you look across the spectrum of countries in Africa where malaria is still prevalent, you will find some of the countries have an effective health system in place. The clinics are paid regularly, the staff are trained, and the work is done. But these are not always present. And sometimes, there's a change of government and a change of interest at the local levels, and the staff have changed. So you need a consistent way of approaching it.
Yeah, just another more scientific question, and for you, Clive, and then I have a few more for Bruce. But if we use the ACTs, if we use the insecticides, are we seeing resistance? I think I know a little bit about this as well. But can you talk a little bit about the resistance patterns of malaria with the use of insecticides with ACTs, and where do you see the kind of the future of worries about, if there is any, around resistance?
Oh yes, the resistance will develop, but it's also interesting that now in Zimbabwe they've gone back to DDT, because the mosquito population hasn't been exposed to DDT for something like 50 years.
Oh, interesting.
And they've lost their resistance. So, but that's part of the scientific work that's got to support it. Is when you start to detect resistance to a current insecticide, then you have to look to see what's still susceptible.
Yeah,
and that involves a pretty good, a good basis, scientific basis in the area to be able to assess that.
that's good. And I think we're seeing same with the drugs, the ACTs as well. Resistance patterns.
Oh, the drug is, yes. I mean, we have two problems here. We have the drug to treat, and we've got a vector that's involved. One of the big problems with malaria now we're finding in Zambia is asymptomatic infections. So people are walking around carrying the parasite, but they're not sick. They don't go to the clinic. They will come down occasionally with a headache and parasthemias, but the parasthemia doesn't last long, and by the time you go to the clinic, your immune system may have pressed that out, had eliminated that group. So
hence the need for continuous development of new drugs.
So that's why it's so important to have a drug approach plus a vector control approach. And I want to mention a point that Bruce mentioned the other day, earlier on, with schistosomiasis. We've got a very good drug for schistosomiasis. Bilharzia is a much easier word to say, but anyway, we're in America, we say schistosomiasis. But the parasite's transmitted in the water by snails, and they do nothing. I mean, I've seen papers recently published that have ignored completely the fact that people use the water for their source of drinking water, their bathing, and all of that. And they go around treating them all with praziquantel. Well, you cure the person, but they walk down to the river and have a drink. It would put their feet in the water, and they get reinfected because the snails are there. So the vector is so important, even when we've got good drugs.
Bruce, so I guess similar question, but we knew this was kind of going to come up. But Henry Wasserman asked, "What is the mechanism of action for ivermectin, and why did anyone even thought it was possible to use against COVID?" So I know you're ready for this one. So
yeah, well, so the exact activity, how it paralyzes the parasite, prevents the parasite from consuming food to stay alive, basically. It's a good question about whoever thought this would work against COVID. Some Australian in March 2020, they did a study that showed that ivermectin, a large quantity of ivermectin, would kill the virus in a laboratory in a petri dish. However, it was 2,000 times more powerful than the human formulation given for onchocerciasis. It would have been very toxic to use for humans. And then there were these studies that suggested that ivermectin might be effective against COVID. However, there were a lot of problems with the studies. There was one in Egypt where was actually withdrawn because it didn't publish the underlying data. There were a lot of questions. It was withdrawn. The one in Argentina the one in Egypt had plagiarisms. The one in Argentina had fabricated data.
Was there something in the underlying mechanism of action would elicit someone to hypothesize it would work against the replication of the COVID virus? Was there any?
I don't think so. I don't think so. And then the largest study that was done, I think McMaster University in Canada, the largest one showed ivermectin had no effect against COVID, and so and I think that's basically been the conclusion across the board now.
And are you seeing any resistance patterns within riverblindness with overuse with use of ivermectin?
Not with ivermectin. There was resistance that developed with one of the insecticides, Tamifos, which was widely used under OCP, and insecticide developed and spread throughout the whole area, and it almost brought the program down. But the program had operational research going on and developed backup insecticides, which were used in rotation, and then eventually the resistance problem was completely reversed, and now Tamifos is being used again. But the major insecticide being used is Bacillus thuringiensis, which is a crystal that exists everywhere in the environment. It's benign for the environment. It's approved by EPA for gypsy moths in the eastern United States. So, and that's the one that was used most extensively after the resistance problem developed.
Okay. And another
to that
question, you had mentioned eradication earlier for river blindness. Paul Keating asked a question about, wouldn't the animal reservoir prevent the elimination of onco?
No, there's no animal reservoir for Onchocerciasis, which is the parasite for humans. There is a similar parasite in cattle, but it doesn't cross over. So
okay. Yep.
The Onchocerciasis is confined to humans.
So definitely a good target for eradication is what you're saying. So
I'm not sure about eradication, but eradication like smallpox, no.
No.
But
okay.
Elimination in geographic areas, yes.
Okay, that's helpful. Okay. Another broader question for both of you is, what other diseases are considered neglected tropical diseases, and what current initiatives are in place to combat their spread effects around the globe? Pretty broad-based question, but I think either one of you could jump in for this one. Maybe Clive, you go first, and then we'll have Bruce. Yeah, very broad question. Right?
Well,
the other NTDs. So
yes, so there are numerous NTDs, and the problem about sustaining the control programs is that there's there has to be, it has to be seen by the local people as something that is serious to them. Many of these are fairly, I won't say benign, but I mean, take, for example, hookworms and tapeworms and things like that. These are neglected tropical diseases because people tolerate the infection if this infection is low. It's only when you get extremely heavy infections that it becomes life-threatening. So many of these neglected tropical diseases can sort of maintain an infection potential in the population, but because they don't really make people feel sick, there isn't much pressure on the government to do something about it. I mean, a very good example of that is Chagas disease. Chagas disease is a trypanosome that's transmitted in Central and South America, and even in bits of this country as well. And people get infected, but they really don't suffer clinically from it. However, it attacks the heart, and you get cardiac failure, just like that, and somebody drops dead because their heart has stopped beating or it can't pump anymore. And so these conditions are not associated by the public with the infection. We know that they are, but that information needs to be publicized.
Yeah. Bruce, any of your comments or opinions on the state of the fight against NTDs more broadly?
Yeah, I think last I checked, there were WHO listed 20 diseases as NTDs, but the 5 major ones we now have drugs for, and they're being donated by the pharmaceutical companies that produce them. So it's hopeful. Again, the five are onco, lymphatic filariasis, schistosomiasis, trachoma, and the soil-transmitted helminths. So I think the picture's pretty hopeful if we can get these out on a continuous basis to the populations that are threatened by them. Yeah.
Okay. I mean, go ahead.
I would still say the answer is no, because people, if they don't have toilets, they will defecate or urinate in the water, and then the snails get infected. And people will go and swim in the water, and they do. You only have to touch it, and you get infected again. So there have been a huge amount of something's happened to my.
You're good.
Yeah, something's happened to my computer. It's gone bad.
No, it's fine. Looks like there's load
but you
shedding.
can hear me and
Fine.
you can see me. Yeah, hopefully you can see me.
Yeah, we can see you fine.
It's so easy to be infected, and it's most of these programs where they've done mass drug administration has not eliminated schistosomiasis, and people go back and get reinfected. So you have to deal with the vector. This is what I feel. There's a tremendous effort on drugs, and because it's a panacea, but it isn't going to work. Certainly with schistosomiasis, and even with trypanosoma cruzi. So there isn't a single way to do it. It has to be done not only looking at the infection in the person, but the natural condition as well. I mean, trypanosoma cruzi affects numerous mammalian sources, so it's a zoonosis.
Bruce, any comments to that?
No, I mean, Clive is the expert on schisto. I mean, he would know far better than I would.
Okay.
I think the diseases which two of them, which I think certainly can be eliminated, are onco and lymphatic filariasis, because you've got good drugs. And you've got, the Merck ivermectin And then you've got GlaxoSmithKline with albendazole, and when you combine them, they're quite effective against lymphatic filariasis. So the difficult one is going to be soil-transmitted helminths. That's going to be difficult. I mean, ivermectin works against those as well, but they're so widespread, and they're not. They people live with them, and so not as concerned about getting rid of them. The best way to get rid of neglected the soil transmitted helminths is to treat onco and LF, and they'll die. The soil transmitted helminths will be treated as well because the same work against them. But
I think a longstanding question has been, for high burden diseases, the role of the private sector. And so, Bruce, you'd mentioned that Merck was giving away ivermectin for free. I've worked in this space for a very long time, and I do know, especially in Africa, you need the private sector, but there's often a large degree of corruption between the government sector, the private sector, and then the donor community. I think, very rich countries like Nigeria and Angola have gotten vast amounts of donor funding for health efforts, but don't necessarily fund those health efforts themselves. They don't prioritize them. And you create a cycle of dependency and corruption. Any thoughts on the role of the private sector on having countries having greater, again, ownership of this, and the role of institutions like Johns Hopkins or others in kind of providing a longer term solution to this rather than just being kind of charitable donors? That was a heavy question.
I think, so back, it was 2012 when they had the London Declaration, is when the international organizations, the donors, and the private sector all came together. And that's then what ultimately led to the ESPN program. And so the pharmaceutical companies are much more collaborative now. And I think that has a lot to do with Merck's donation.
Yeah, they were a leader in HIV drugs as well, right?
Exactly. So, yeah. Yes, they were. Merck has been quite active in Botswana with HIV, and
so GSK has. But that's the pharma companies. How about in country with supply chain capacity? Clive had mentioned, the researchers, local researchers, local physicians, nurses, healthcare work, pharmacists, et cetera. Any thoughts in terms of how do you create, again, vast amounts of money have been put into health system strengthening. It often goes into the pockets of government officials in those countries or local private sector companies. How do we manage that so we have better outcomes? So is there a role to play for the development community to strengthen those institutions, governance, as well as, I guess, grassroots on the ground in terms of availability of the medications?
Well, I think what you had with, I know the Onco program, so I'll speak to that. But this was such a large program, well funded, and they made an effort to Africanize the whole staff of these programs. People were trained at WHO, and they then, when the programs ended, they went back to their governments and started functioning in their governments to continue the work that they had done through the programs. And I think that was quite beneficial.
Clive, any thoughts on that?
Well, I know the OCP too. I did a program, a work there with them. OCP was a really an Africanized focus, and it was run by African scientists.
Exactly.
What the problem is that if we send somebody to university, bring somebody over here to university. We've done this with Schisto and with malaria work. Is they do their doctorate and then they don't want to go back home because there's no there's no funding back home. There's not. So what I've been involved with an isomer. The isomer concept of NIH is very good, that you've got to have a center of excellence in the country.
Yeah.
And the only, the American scientists have to put 30% of their time into that program, that in the field. But at the end of it all, they come home, and the isomer, the center of excellence, will collapse in the user country. So what's important is some of the research money that NIH puts into the isomer should actually go to universities in Zambia or in Tanzania or wherever, just to train and employ scientists, so they don't have to wander off to other places.
No, that's good. Is there any advances? Clive, you'd mentioned the use of mobile phones, right, in kind of tracking surveillance.
Yes.
Bruce, obviously ivermectin was a huge innovation. Has there been any? What's your thoughts in terms of current innovations that are occurring that maybe could change the game in terms of NTDs, including maybe some innovations that occurred during the pandemic as well? So just want to get your thoughts on current innovations, upcoming innovations you find to be potentially very highly impactful. So.
That's a very difficult topic to pick up, because really, I've been in Zim recently, about 3 years ago, and in Zambia the same. The difference is chalk and cheese. When you go to Zambia, you go to the Ministry of Health, they talk to you. You go to Zimbabwe, and there aren't staff there anymore, because there's no sustainability.
Yeah. Bruce, any thoughts on innovation?
Yeah, I come back to what I spoke about during my remarks. I think one important breakthrough in terms of getting ivermectin out and addressing and making sure we could stop transmission was this community-directed treatment method, which took quite a while to develop, and it worked extremely well, because you have no institutional structure in the rural areas, which is where these diseases are concentrated. But getting the communities themselves involved, and they had the incentive to step up and get the drug out to their people, and it worked for onco. Now I realize this probably wouldn't work for schisto. I think it works for lymphatic filariasis, and there are a few drugs there you can like ivermectin and albendazole that you can get out through this method. Not so much for schisto, I guess, which is being where praziquantel is being distributed through the schools. Is that right? Clive, if I'm not mistaken.
The problem even in the schools is, I've seen it myself. The kids are going to school. The schoolmasters have got a vegetable garden next to the river, and at the end of this class, the kids have got to get down to the water and collect water and water the teachers' vegetables. That's how they get infected. So this is something that's fairly standard in rural schools that I've seen in Africa, well, in my part of Africa certainly. but
yeah. All right. We have.
You've got to change our whole philosophy. The government has really got to take these things seriously.
I mean, this is my experience, Clive. Is that I was in Tanzania, I've been in Zimbabwe, I've been all 30 countries in Africa. It's when you have a stable government and a willing government, you can make big change.
Yes.
And if you don't have a willing government, you're pouring money down the sink. And
Yes.
we've done this again and again. And so I think the partnerships with the government is one of the most crucial things that we can foster. We got 2 minutes left. Just maybe closing thoughts on the role of Johns Hopkins. It's had such a huge impact on global health overall. But what else can institutions like Johns Hopkins or individuals who joined in today help with your cause, with public health in general? Very broad. Last question for
Well, yes, I think we can, and I think that we have a moral obligation to train these people and provide the expertise that they need to be able to take home. I know that we've supported through the Bloomberg Trust a very big research station in Zambia, which has has MDs and PhDs running it now, all Africans, and it has a huge potential, but how to sustain it, that is a question that we can't solve here.
Yeah,
that's how I see it. Yeah.
Bruce, any final comments, and then we'll close, and thank the audience for joining.
No, I think I do think that the universities like Hopkins, that has its expertise, it's imperative that stays involved in these diseases. They don't exist in the developed countries, so but if we don't lend our expertise in the developed world and in the universities that has an expertise, it's not a complete package in pursuing solutions for them.
Right. Good. Look, Clive, Bruce, thank you so much for your contributions to global health overall. Thank you for your time today. We had a really good audience participation. Thank everyone for joining us and being patient with the late start. But with that, we'll go to a close. And thank you, everyone.
Thank you very much. It was a pleasure.
Thanks very much, John.
Yeah.
Thank you.